St. Patrick's Day. This is the
Happy St. Patrick's Day from the St.Louis Fest.
закончился парад Святого Патрика, который сегодня я отпраздновала, и здесь было много всяких бус. Эта станция называется Union Station. А в треке я здесь никогда не была внутри. Оказывается, здесь очень даже красиво. Это магазин кардиналов, бейсбольной команды Сент-Луиса. Вот здесь так очень даже уютно, красиво, необычно. Здесь магазины, вот там шляпы такие типичные, американские. Это внутри всякие магазинчики здесь. посмотреть, как у нас на работе. Он прямо вот он делает, а потом нравится еще попробовать. Можно и купить. Да, товарищ прям чудеса делает с этой штукой. Покатили. Здесь такой беленький нарезатор, что кусочки. Непонятно, это какая-то сладость или тесто. Еще все продают. Надо попробовать. Сладкая штука.
You're welcome. Hey, youngster. What's going on? Yeah, I seen you out.
Типа на 100 рублей. Один похож на фудкортер. Как всегда, американцы жить не могут без еды. Вот тут народ после парада отрывается. Стоят в очереди, уже интересно, где-то Union такие перекладины. Это всякие значки по случаю дня Святого Патрика. Надо купить. А вот это, я нахожусь снаружи, вот это Union Station. Здесь такое озеро, оказывается, приятное. Крыша такая необычная. На той стороне находится Seafood House. И вот здесь даже играет какой-то ансамбль. Народ после этого празднества, после парада дня Святого Патрика, релакс. И даже вот там есть хот-дог кафе. Надо же, я для себя открыла новую страничку в Сент-Луисе. Вот это хот-дог кафе. Надо сюда обязательно сходить с кем-то своей знакомой. Это такой мост. На ту сторону Seafood House тоже не мешало бы посетить.
I'm happy to see all of you at my presentation today. The topic of my presentation is mass-spectrometric mapping of GABA-A receptor. At the beginning, I would like to tell you about very interesting protein, GABA-A receptor. Can you imagine that such small, tiny molecules can be very responsible for brain activity, and they can be responsible also for brain disorders. They are very important for learning, concentration, for memory, and they can be modulated by different pharmacological compounds. As you can see here, GABA receptor located in membrane, and they can interact with different membrane proteins. In my presentation today, I'm going to focus on three the most important and interesting points. At the beginning, I would like to tell about you immunopurification of GABA-A receptor from small amounts of brain tissue. The second, I will tell you about mass spectroanalysis of primary structure of GABA-A receptor. And finally, I will explain you how to use this mass spectrometry analysis to identify neurosteroid binding sites. If you have some short questions, you can ask me during my presentation. And please save your long questions to the end of my presentation. I will also be available for informal conversation after my talk. So now we come back to my presentation. At the beginning, I would like to tell you about features of GABA-A receptor. Everybody knows that this GABA receptor is a major neurotransmitter receptor in brain. And several subunits of GABA receptor have been identified, including delta, theta, omega, P subunits. But the functional complex of GABA-A receptor contains from 2 alpha, 2 beta, and 1 gamma subunits. I told you already that these GABA receptors are very important for learning, memory, concentration, and they are target of many clinically important drugs useful in the treatment of anxiety, epilepsy, schizophrenia, sleep disorders, and some other brain disorders. As you can see on this slide, the GABA receptor has specific binding sites to bind GABA, benzodiazepines, hypertoxin, barbiturates, and steroids. Most ligand binding sites are already identified, but the localization of neurosteroid binding sites is still not clear. Now the identification of neurosteroid binding sites is target of investigation many scientists in the world. And you can ask me why we are so interested in investigation of these binding sites. Because neuroactive steroids, they can be sedative and neuroprotective agents. They modulate the function of GABA-A receptor and can be natural anesthetics. And a major challenge in pharmacology now is the discovery of these neurosteroid binding sites, because it will allow to design many new therapeutic anxiolytic drugs to treat brain disorders like major depression, epilepsy, schizophrenia, alcohol dependence, and some other diseases. So the major goal of our research is to identify the neurosteroid binding sites of GABA receptor by mass spectroanalysis and photolabeling, and characterize the post-translational modifications of GABA receptor. The characterization of post-translational modifications is also very important because it's allowed to understand the signal transduction in central nervous system and understand the regulation of GABA receptor activity. So now I'd just like to tell you a little bit about mass spectroanalysis. The application of mass spectroanalysis in the last 10 years has been extraordinary, because this mass spectroanalysis is very efficient tools, and these tools allow us to identify primary structure, proteins, receptor, analyze post-translational modifications, including phosphorylation, acetylation, glycosylation, and some other modifications. And also, this mass spectroanalysis allow to identify ligand binding sites by comparison the labeled and unlabeled samples. So now there are 4 advantages to using of mass spectroanalysis. At first, the high sensitivity, the second, the rapid speed of analysis, the large amounts of information which you can get from the just one experiment, and as I told you already, ability to characterize post-translational modifications. So, now I will tell you about immunopurification GABA-A receptor from very small amounts of red brain tissue. We used about 10 milligram red brain membranes, and we solubilize this with a specific detergent, desoxyfolate. Then we loaded this lysate on protein in agarose column immobilized with anti-GABA receptor antibodies. It was complex of different antibodies directed against alpha-1, beta-1, beta-2, beta-3, and gamma-2 antibodies. I would like to note that these antibodies have been kindly provided from Vienna University, Professor Werner Ziegler. So, after immunopurification, we eluted by glycine about 6 picromole GABA-A receptor. We measured the amount of GABA receptor using RO15 assay, which allows to identify binding sites between alpha-1 and gamma-2 subunits. So, after this step, sample has been concentrated on centricon plus membrane and then submitted for nano-safety meta-analysis. So, we loaded these samples at the beginning at the 10% gel, and this gel has been analyzed by Cipro Ruby and by Western blot. As you can see here, Cipro Ruby staining shows very major band, around 52 kilodalton, and on Western blot, this band completely corresponds to cross-linking reaction with antibodies for gamma-2 subunits. So, this both data shows localization of GABA-A receptor subunits in this position. All this major band, 52 kilodalton, has been selected, and 8 spots has been excised, and they have been submitted for mass spec analysis. So now I'd like to tell you about nanospray ionization, this nano-safety meta-analysis, just very shortly. If you look at these slides, you can see operating principle of nanospray ionization. Usually, acidic aqueous peptide solution is sprayed through the small needles, about 5 micromillimeters, at very high voltage with low flow. And ionization peptides take place, and multiply protonated peptides ion absorbed. On the first step, these peptides analyzed according to the accurate mass and charge states. And then linear trap performs an SMS fragmentation analysis using collision activation. On the next slide, you can see actually this ion trap Fourier ion cyclotron mass spectrometer, which we used for analysis our digestive mixture. So, analysis of spots 3, 4, and 5. In our case, using nano-safety method, definitely identifies the presence of alpha-1, alpha-2, alpha-5, beta-2, beta-3, and gamma-2 subunits of GABA receptor. But our initial work has been focused on obtaining sequence of alpha-1 and beta-2 subunits. So, nano-safety meta-analysis is pretty complicated analysis, but I'll explain you using these spectrums. For example, here you can see selected ion chromatograms of VKDPL peptides from nano-safety analysis. Predicted molecular theoretical mass for these peptides was 4067663. According to our experimental results, we found accurate mass. You can see here 4067663 7663, and this molecular mass produced a tandem spectrum, and difference between these two masses is 0.5. This value shows that these peptides exist as a double charge peptides. So we calculated delta. Delta is the difference between theoretical predicted mass and our observed mass. And then we calculated tolerance. Tolerance 1.7 ppm is less than 5 ppm. So this data shows that probably this peptides has sequence UKDP and RK. But we need to confirm is this true or not. So we did the next step of analysis, and it's called MSMS fragmentation analysis. MSMS fragmentation analysis means that fragmentation peptides ions induced by low energy collisions. And during this fragmentation, peptides cleavage of different amino acids. Each amino acid has a set of B and Y ions. And these ions allow to reduce the sequence of peptides. So we did sequence peptides using database search, mass spec program, and also each sequence peptides has been manually confirmed using Excalibur program. For fragmentation ion spectrum, we compare it with theoretical spectrum derived from the protein prospector program. For example, this slide shows an SMS fragmentation spectrum, the same peptide, and you can see that during fragmentation process, all these ions should be absorbed from Y1 to Y6, and they will confirm sequence peptides in this direction, from the right to the left. If we'll see ions from B1 to B6, they will confirm sequence peptides from B1 to B6 from the left to the right. All these ions, especially from Y1 to Y6, has been identified. And we'll also be able to identify almost all B ions. We just did not see B1 ions because it has a very, very low molecular weight. So this data definitely confirmed the sequence peptides what we found. I would like to note that all these peptides have been analyzed according to the accurate mass and isotopic distribution and MSMS fragmentation analysis. In the end, we identify peptides for alpha subunits of GABA A receptor. Different colors shows a different charge state for peptides. Single, double, triple, quadra, penta charge peptides. First column shows the sequence domain which we identified. For example, we identified N-terminus domain about 91%, and M1, M2, NM3 linker, and also transmembrane domain M1, M2, NM4 linker. So if you look at this part, you can see theoretical compared with observed mass, and all these masses have a tolerance less than 5 ppm. Tandem analysis shows that some peptides have tandem. You can see yes, yes, or some peptides no tandem, because it's not always possible to identify tandem spectrum for all peptides, because some peptides have very, very low abundance. And in this column you can see verification found for all these peptides. For example, phosphorylation, methionine oxidation, carbamide methylation, and I would like to mention that in most cases phosphorylation has been observed in soluble part, in N-terminus. Based on this data, we calculated coverage of alpha-1 subunit systems. And coverage for alpha-1 subunits was 81%, definitive tandem identification 47%, tandem consistent identification 26%. Tandem consistent, it means that we would be able to see many ions, but not all complete P and Y ions, about 60 or 70%. We also calculated coverage of alpha-1 subunit domain based on results of non-swiftness analysis. For example, N-terminal has been identified on 85%, M1 domain, M2, M3, and M4 domain. They are transmembrane domain. And you can see that we were not able to identify just M3 domains. And I will tell you about this later. Why not? Linkers, they are usually soluble parts, M1, M2, M3, M4, and M and C terminals. They perfectly identify and they give about 100% of identification. So, based on all this data, we designed a map for alpha-1 subunits, GABA receptor from red brain. And this yellow color shows all the amino acid residues which we detected and determined by mass spec analysis. And red color shows uncovered sequence. You can see, but this is soluble part. It's N-terminals. It's identified on 85%. Such small pieces like MK or these two not possible to see by mass spec analysis. And for example, these huge large peptides also not possible to identify because there are no cleavage sites for trypsin here. But I would like to emphasize that we identify this M1 and M2 domain. They are transmembrane domain, and usually, and M4 domain also has been identified. Usually this transmembrane domain, they are very hydrophobic. They are sticking to the sides of polyacrylamide. They are binding. They are not so easy extracted, and usually it's very difficult to identify the transmembrane domain. I'd like also to explain a little bit why we did not identify this part. If you look here, there is also huge peptides at cleavage site here, because trypsin cleavage amino acid residues between arginine and lysine. So it has about 6 amino acids, too big. But we perfectly identify this M3 and M4 linker and C terminal. I'd like to tell you that the same analysis has been done also for beta-2 subunits. And in case of beta-2 subunits, we identified about 79% of total sequence coverage. Here you can see mapping of beta-2 subunits GABA receptor from red brain. And you can also see that we identified partial of M1 transmembrane domain, perfectly identified M2, M3 domain, M2, M3 linker, and here we identified M3, M4 linker. This part is very interesting. If you look here, you can see that there are many cleavage sites for trypsin. It can cleavage in this position and this position, and it's produced very small pieces which you never can see by mass spec analysis. Probably we need to use another protease in such case to identify all this M4 domain. So I'd like now just to emphasize the results of mass spec analysis. We identified high sequence coverage for alpha-1 subunits, about 81%, and for beta-2 subunits, about 79%. We also be able to see some post-translational identification, including modification of subunits, phosphorylation, methionine oxidation, modification, they all have been characterized. So, now just I'd like to tell you that today I told you about the modification of GABA-A receptor from small amounts of red brain tissue. And also I told you about mass-spec mapping of alpha-1 and beta-2 subunits of GABA-A receptor. I can make some conclusions that nodal CFTMS analysis is a very powerful approach which allows to identify primary structure of low abundance neuronal ion channels and characterize the post-translational modification of proteins. And it also allows to identify neurosteroid binding sites. I will tell you about this in my next presentation. So, our future plan includes characterization of neurosteroid binding sites GABA receptor using mass spec analysis approach in photolabeling, and study the molecular mechanism of regulation of GABA-A receptor. In the end, I would like to tell thanks all these people, Professor Alexeevich, Professor Vitauza, Professor Ziegler, and some people from their labs for contribution in this research. And thank you for your attention. And now I will available for you for your questions. If you have some other questions, please don't hesitate to ask.
I'm not sure I understood exactly what you said, but we already have whole genome sequence of human or other organisms, so I think we can predict the protein sequence based on the DNA sequence. So your work is identify the protein sequence of GABA receptor isotypes by using mass specs. So I'm wondering what is your main purpose of mass spec?
Okay, the question was what is the main purpose of this research? Why we are doing mass spec analysis of GABA receptor? Because it's already we found from cyclone DNA, right? So the idea at the beginning, I told you that we want to use this mass spec analysis approach to identify neurosteroid binding sites. So unfortunately, I have no time to tell about this, how to use this approach. But if you compare the sequence labeled and unlabeled protein by mass spec analysis, and you can find peptides which contains your label, then you can definitely identify amino acid residues which binding with your label and identify these ligand binding sites. This is why we are doing this mass spec analysis. So, do you have any other questions? Okay.
I want to prepare a peptide library, and maybe I want to prepare 10, but sometimes we only get 9 or 8. So can we use the mass to detect the sequence of the peptide?
Could you repeat question again? More clear and more shortly.
I want to prepare 10 monomer.
You prepare it for your research.
N equal 10. 10 amino acid to prepare 10 peptide. But sometimes only get 9 or 8, and some missing. And so can we detect which one is missing by using mass? Thank you.
So you have a protein, right?
Yeah.
And you detect some peptides, but some peptides missing. You don't detect them. Right?
Some amino acid in the peptide miss.
Some missing. Yeah, I think you can able to detect this using nodal CFTMS analysis, because you can even also see post-translational modification. It means you can find which amino acid has a specific substrates like glucose or phosphate molecules. So it's very sensitive methods. Okay.
Thank you.
Thank you for your question.